Okay. So today is day 4th, and we got so many things we passed through. I think something, at least 10% if you remember, that's good. So I'll continue this Henipa virus, and yesterday we finished the Ebola virus, which is BSL-4. So this is the Henipa virus, is BSL-4 also. So in Henipa virus, there are a lot of viruses. You can see the list here. Hendra, which is originally in 1994, has been discovered, and then Nipah virus. Like, so the Nipah virus is 1998. You can see there are so many other viruses there. But the more important we are talking is the Nipah virus. Why? Because it's affect the humans. But you can see those viruses are structurally similar to paramyxovirus. We talked about paramyxovirus, maybe not. I skipped the measles talk because there is not enough time. But if you anybody interested on listening or getting the measles informations, because measles another thing, there are some outbreaks happened in the United States. So I thought it's kind of WHO is still thinking whether it can be as a re-emerging disease or not. So that's why I have not presented, but it's the same family with the where measles is. It's in the paramyxovirus. It's a negative strand, single-stranded RNA, and it is the largest paramyxovirus genomes. And this is the schematic structure. You can see lipid envelope, nucleoprotein, phosphoprotein, matrix protein. The more important is the glycoprotein and fusion proteins. They are the structural proteins. And inside the RNA is there. And RP is RNA-dependent RNA polymerase. So here is a more functions. Several talks we give, like last 3 days. I always say multiple times, like virus is smart, right? So where from smartness comes, where the brain is, right? So these are the proteins are the brains. If you see the functions, the N proteins, they protect viral genomes. P proteins, they interfere host immune. So they have the all kind of defense mechanisms. Those proteins, that's why they need those translation and to hijack those functions, okay? So M protein provides structure and protects nucleic acids. Fusion also facilitates virus entry. So they have their protein has their functions, and they modulate the host functions. So that's and then say AL protein, which is the biggest one, which is actually making copies, negative sense to positive sense to negative sense, because we need for replications. Tissue tropism is an infection through respiratory mucosa because it comes through respiratory tract, also by oral tract. So I'll tell you how it transmits. Tissue lining oropharynx, because through orally it goes to the pharynx, or respiratory tract, or nearby lymphoid tissues. So nearby tonsils and all those are lymphoid. So take through the tonsil first. Through nasals, there are local lymph nodes, they goes. Secondary replication, amplification to the vascular epithelium leading to disseminated. Again, we talk about DICs, right? It causes hemorrhage too. So it affects those vascular epithelium. And that's why endothelial infection, ulceration, thrombosis, vasculitis, those are multiple things happens. The major thing I'll point out, the lessons in CNS, it goes to the brain also. That's why it causes encephalitis. We are not talking Hendra. Hendra goes to the lung, but these viruses also go to the lung. But major issue happens because it goes to the CNS, and other systemic infections happen. So this is a timeline when virus has been evolved. 1998 in Malaysia, first time it happens, they find the febrile illness mainly in pig farmers. Those pig farmers they get infection, those infected, and they are showing the fever. And same thing happened in 1999. Then, because based on the village where they found, like one of the villages like Sungai NIPHA, so then the virus has been named as NIPHA. And I told you, like whoever discovered first, they say based on their name or some country or that kind of idea is, or the whoever discovered by their name. But it rarely happens because it is not like somebody has discovered. It's based on where it has been finded out first time. So. And you can see this is the trend happens from 2001 to still to has been discovered in Bangladesh as well as India. And there are a lot of outbreaks happens mostly in the Kerala, and you can see those Ernakulam and Malappuram, those areas. It has a lot of outbreak happened. At one point, like 2023, there is a lockdown has been imposed to control those transmission. Bangladesh very severe. I think the second worst outbreak happened 2011. But it is a very seasonal. Every year in India as well as in Bangladesh, this outbreak is happening. So again, another way to show that, you can see India, Bangladesh, Malaysia, some part of Philippines, and you can see here how many deaths, always more than 50% death is happening in those areas. So it's very fatal disease in that aspect. And Bangladesh also, you can say that's near the Dhaka area and Rajsakhi and Rangpur, that area, there are a lot of outbreaks happened. And you see that table shows here, there are how many cases are there, how many deaths happened, and CFR is the, how much the cumulative case fatality rate actually, case fatality. So how much is 100% death that time, but there is no death. So, but it's like very fatal. So this is the mode of transmission, bat to human. So what happened? The fruits are there. All the bats are fruit eating, right? So they are eating and the bat already has a lot of reservoirs of different viruses. One of them is NIPHA. So they are eating there and they are releasing their urine or fecal matters on those fruits, and fruit down, and the pigs are eating, and you are giving a hug to the pigs, or you are a pig farmers always in close contact with the pig, he got infections. Another, we all know the palm tree, we get the soups, right? The palm soup. So, go there, but it is not. Then the bat put, the urine or fecal samples may go there, and you're contaminating that, and it will be consumed by everybody, and then that's where the transmission is there. The human-to-human transmission happens in Bangladesh also, but it's like only, very rare cases, but it is there. If I am infected, and if my secretions or blood comes close to you, it's some like Ebola. There's a chance also human-to-human. So these are the bats which are mostly responsible for the pteropid family, and there are different pteropid families are present in different country. Those are the species name, and you can see those are actually responsible for transmitting those, and they are all those areas. So this is basically the transmission. So they are the natural reservoirs. Nothing happened to them, okay? But they also can be transmitted, these species. And some of the species like monkeys we studied in depth, but all the species can be infected by Nipah virus. Nipah virus also has been transmitted, so somehow the hearts got infected by maybe their secretions, saliva or fecal samples or urine, and hearts also can get infected, but it's mild severe. Another is pigs. So basically human got from all those sources, directly or indirectly from those animals, they got the virus. So incubation, no need to say what is incubation is. It's between 7 to 10 days for pigs and less than 2 weeks in humans. And it's very fatal disease. Infection to disease ratio is 85 to 92%. It's non-specific fever, CNS involvement. So it's go to the brain. So you can see all kind of CNS-related symptoms like headache, dizziness, and vomiting, loss of consciousness, those things. This is very important. See, in Bangladesh, strain very virulent. This may cause 70%. And the survivors are like 75% fully recovered, 24% has the neurological signs because it will go to the brain. So it is a kind of permanent and that relapsing encephalitis. So it may come at some point. You may see the persons who are behaving well, but sometimes it comes back again. So you have to do those kind of. So it's a very busy slide, but you can see the overall is there are a lot of assays has been used. Lateral flow device, I think everybody knows now. This is the same kind of techniques we have done for the COVID, right? Put your nasal swab and put it there, and you can see those lines, positive or negative. So the lateral flow assay has been developed for this. Loop-mediated, we'll tomorrow loop-mediated isothermal amplification method, sophisticated PCR method. RT-PCR also house, the grain, copies there. TACMan also you can. So both are kind of similar. Multiplex PCR has been used, and also all those ELISAs, Luminex based. If there are anybody interested, Luminex, we did a lot of work on Luminex, so you can discuss. But Luminex is also you can. It's a bead based assay. You just tag the antigens on the top of it and detect it. So it's very sophisticated, very sensitive, very expensive too. See, that's why it needs advanced technology and expertise. And virus neutralization and virus isolation is an old but gold technique and it can be used for it. So the best way to prevent it is reduce bat and pig contact. How? Make cages, washing those things, don't keep close by trees, make sure there is not bat colonies growing. So that's the kind of housekeeping things here. I mean, if you don't keep fruit trees close by, then bats will not come. That's kind of, so cut down trees, don't put any kind of fruits there. So that way is the kind of preventing the close contact with them. And improving hygiene and monitoring pigs. And for like, as you know, the collecting when you like sap, palm sap, you just cover it. So that, bats cannot go in to drink those because those are actually, food for them. So that way, those are not contaminated and you can drink. So before you drink, make sure they are not contaminated with this. I mean, that is the major transmission happens in India and Bangladesh. They are very tasty actually, right?
How are we getting it from the pig?
Huh?
You said by being close to the pig.
So pig, what happens now? So pig, those pig farms, they have like fruits. So those are fruit-eating bats, right?
No, how are we getting it from the pig?
Oh, because you are, close to that. No, it's not. It's a close contact.
Close.
Yeah, either pig is licking on you or do something, and it comes to you, or it also comes through the urine to the fecal samples. If you are not taking proper care of your body, then you may go close. Yeah, that's happened. So there are timelines. The vaccine has been developed. I mean, trial has been developed. So far there is no vaccines. So you can see there are since 1994 there are a lot of vaccines. The Hendra virus have these vaccines, not for any vaccine. The Hendra virus equine vaccine has been licensed. That's for equine. But for Nipah virus, there is no vaccine. There are different animal models has been tried. There are preclinical vaccines, VRP. Actually, we worked on which one? I think, yeah, this one. We are working with this one with CDC. At some point our publication will come out. But these are the vaccines has been tried also mRNA and VSV vector vaccines. But yeah, this is, we finished, I think it's very good vaccines we come out. We finished that, we are working on the paper now. But this is the stage how the vaccines discovery is still going on, but there is no commercial vaccine available. Yeah, so CDC vaccine, we did VRP, lacking F gene. It is also very nice vaccines, but there are other companies did this, as well as in the different universities. Moderna has those vaccines, but all are very early stage of this clinical trial. Okay, I'm switch gear, but just to show what research we did also for NIPHA. Okay, it's little maybe go over the head, but just get some basics of it, right? Because I like to give the new touch of it within the new technology. So the emulate organochip, okay? So it's like a chamber here, there is a plate here, and there are kind of connectors here. So these are the reservoirs here, and it is a vacuum channel, so it covers it, and there are outlets, okay? So what happened, basically the fluid goes in here, and then come out and go back. So it's a fluid comes in, pass through this, and same like here, it goes through and come back, okay? The two chambers are here, and the fluid is passing through. Basically it is, okay? We can make that like a lung model. So for in vitro lung model, how we can generate. Okay, normally we do like a cell culture, which is like a two-dimensional, right? On a plate, we are putting the cells, which does not mimic what is in our body. Then people try to the 3D, which is also okay, but it does not actually express the conformation of this, and also it does not include what different, in blood there are different cytokines, chemokines, and how the cell to cell interact, because that cells only one kind of cells, right? But in the lung we have multiple different cells. So that's why this is the way we did. The top channel we put some cells, and the bottom channel we also put some cells. And there are two vacuums, so we can squeeze it. There is a membrane there, which also kind of has a porosity. That means the cell also can pass through if needed. And this is just to mimic how the epithelial and endothelial, so epithelial and endothelial, just to mimic how the lung is, and we can do the same thing here. Okay, so what happened when air goes in, then it's stretch, our lungs stretch. And then when air goes out, our lungs squeeze, right? So the same principle we can also apply here by squeezing this and putting the stress, putting the air pressure. That's why it has like a vacuum channel, so we can control the same thing as happening in lung. So the idea of this organochip, I mean, if you look over literatures, anywhere, that's the idea, is to mimic the same thing what is happening in vivo, make it is in vitro or ex vivo. Okay, but here we are just focusing on two different cells, epithelial cells and endothelial cells. So this is the instrument. Okay, there's like a nice globe here, and this is the instrument where those, I saw the big picture, but actually they are that size. So this is the chamber. It has different nodes there, and the plate is right here. And you can program it, and then you can design how much, exactly you can put the program like how much lung expansion happens and reduction happens in human. You can set it up exactly same way, and you can put the air. So it's a liquid air interface there. So as as a lung, we have the blood is going, air is going. You're putting the same way here too. And we are only playing here two cells, epithelial and endothelial. Lung has other cells too. It has lymphocytes, a macrophage, that will make more complicated. So we just started with only two cells, epithelial and endothelial. So these are the reasons, because it is very used nowadays. It's a new technology. People are used for drug toxicity testing. That's the major called also to in two-cell mediated interaction on the system. So we used in GBSL4 format, and this successful or not. So this is we did actually. So we understand that this is the chips. There where your top layer you're putting epithelial and bottom doing the endothelial. So top channel and bottom channel. It's the kind of schematic here. The air is going in, the fluid is going in, and there is also fluid going in the bottom chamber. And the PDMS, it's like a silicone-like membrane because we are squeezing, right? So we need something flexible membrane that can squeeze. It cannot be like a plastic plate. Then it will break. So they put the silicone membrane so also the cell can pass through. And here we put different virus. We put the NIPHA virus. We put the TNF alpha, cytokine. We want to see the cytokine effect. We also tested different drugs. You probably know remdesivir, mostly used for COVID, and looked over different other things, how does immune cell infiltration happens, what the barrier functions. So we did bunch of passes, like the bunch of passes we did, all those RT-PCR, plaque, and barrier functions to detect. We can see the infection happening or not. After infection, what is changes happening in those cells. If the change is happening, whether there is cells are moving from bottom to up or not. If there is change happening, what happened to the cell structure, what is happening to the cytokine level. So all those we have studied. You're okay? So this is the model we did to understand. So that's kind of understanding more in depth after Nipah virus goes to the body, as is said. And from P, we can go through, through orophageal, the virus goes in and go to the lung, and then what happens? So this is a little in-depth study after virus goes in, what changes happen. So we saw infections, there is increase, and these are the plaques. I say, you talk about like how you can see they're like a round thing. So this is called plaque. So they count it, how many plaques they form, and then they presents as how many viruses there. So only one virus can make one plaque. Okay. So then by the plaque assay also you can say how many plaque forming viruses are there. We can say these are then you count it and say this many plaque forming viruses are there. This is a Vero cells, which is African green monkey kidney cell line.
This is not in that.
It's not in that. It's just a secondary. So if you go here, we collected everything, I said, right? We collected supernatant. So we collected supernatant. Now we are testing on the output of that machine. And then measuring, so okay, then you saw after giving virus, there is replications. And so this is apical effluent and basal effluent. So what is in the top layer? What is in the bottom layer? So we collected both secretions, right? So then we are measuring virus replication happening in the top, how much is the bottom.
So you're talking about, in a larger sense, you're looking at how
infection happened. First, you have to confirm, right? Yes. First, both epithelial cells as well as endothelial cells, because top is epithelial, bottom is end. So, but because you have to first confirm that really infection happened or not. So this is kind of a confirmation that okay, there is infection. Then so it's there's we also tried different drugs. You no need to worry about. And also, so another thing is infection to see. We talk about CP, cytopathic effect. So this is kind of cytopathic effect. So you can see mock means there's negative, there is nothing. You can see very nice cells individually, but you can see there's a cluster of cells. So this is called, the cells are all combined together. This is called syncytia. Syncytia means they make a big clump of cells. Finally, they will die, but they always, that is the feature of this syncytia. Or somehow the cell wall got additions all together and they make a bigger cells. And that's called syncytia formation. And that also been confirmed with the Nipah virus glycoproteins. We did immunofluorescence microscopy and they are present. So it also, that means it shows, basically those two slides saying, we put the Nipah virus, the virus is replicating, here is the virus is present, we saw plaque forming cells, and we also saw syncytia formation, so virus is replicating.
What is donor?
It's a two different donors we tested, right? It's not like because it's all human cells. So we got the cells, epithelial, endothelial, these are all from human. So you got several donors. And we take the epithelial cells and put that. So it's a donor. Huh? Mock is the control. Mock is the control. This is the infected. And then this is the march, means we march together. So the glycoprotein is here, it is a green color. And you can see the blue. Blue is the nuclear marker, which is, we normally use like a, I think is a DAPI, DAPI. That's the stain, actually stain the nucleus. So when you march those two, you can see this comes like little yellow color. So it's a blue. This is only the green color. And then when blue and green comes, it's little like yellow color. So that means those positive cells are also nucleated cells. So the virus is replicating in the nucleated cells. DAPI is the marker of nucleus. Every cell has a nucleus. That is a kind of confirmatory test. If you see only this, but there is, I don't find any nucleus, that means it is a non-specific binding. It means it's a wrong stain. Somehow you got a stain, but it is a artificial. I don't believe it. Yeah, it is somehow, you are not expert enough in microscopy, you generated some autofluorescence. We call always autofluorescence. That means it's a non-specific.
It's a confocal microscope.
Of course, yeah. But it's a confocal, yeah, that's called microscopy technique called confocal microscopy. Is it clear? So this is a nice video I'll show you. So here what we did, we went a little further. So now we know that, okay, we can infect those organochip. So what we are doing, monocytes now. We put CD14 monocytes. CD14, again, CD14 is the cluster differentiation 14. So CD14 is the marker of monocytes. If you see, like, if at some point you gave your CBC count in the hospital or any doctor's office or somewhere or diagnostic center, if you see the report, they'll put lymphocyte, monocyte, neutrophil, platelet count, everything they listed. How much WBC percentage, all those information is there. So it's the same thing, like when they talk about monocytes, how many monocytes they have, we use CD14 as a marker to monocyte. So, monocyte, when they do, like, when you give the blood and do the CBC, they do based on their other structures and as well as mostly based on the structure, the machine decide those. But here we mostly do like flow based. So we use the CD4 as a marker and then define those markers. So what we did, we sorted those cells. There is a sorter machine. We sorted those specifically monocyte cells and then do this experiment. So this is the specific pictures showing how we did. So you can see the air liquid pose, then cells are going through on the top cells. We put the virus there, and then also we put some neutrophils here, which is, we put neutrophils. And then what that does, the neutrophil activates the ICAM. So neutrophil activates the ICAM, and then it goes internalized on the transmigrate to the upper layer. So what we can do, you can put those top layer. So you put in the bottom channel, right? I put the bottom channel, the neutrophil, and I can find in the top channel the neutrophil. So why did it happen? Because the cell migrate from the basal layer to the upper layer, right? How it happens? Because the TNF alpha is there, which activated, and also neutrophil also went there, it activated VCAM. sorry, ICAM. So, and then made a pore, and it moved there because there is a TNF alpha. So TNF alpha is an attractant of the cells, because when there is, that's why when there is a TNF alpha there, neutrophil goes there, monocytes go there, because they thought, oh, there is something happening, we have to help, because there are a lot of TNF alpha. So that's why migration happens. Then we measure, okay, how much neutrophil infiltration happened on the epithelial layer or endothelial layers. And we have different markers. MPO is a marker of neutrophils, and we detected those. And also we are also measuring how much Nipah virus to make sure that virus is also there, as well as neutrophil marker is there. So also we made different cytokines, basically, and different cytokine stimulators. So no need to go more details, but just to show that organochip is another model where you can mimic what is happening in the human. So what we did, I'm summarizing, okay? So what we did, we made an organochip model. We put the top layer epithelial cells, bottom layer endothelial cells, put the virus on the epithelial cells, and the bottom layer we put the neutrophil. And want to see when the virus induces inflammation, and also put TNF alpha, that is a marker of inflammation. So when there is inflammation, the neutrophil goes in from the bottom to the top, okay, and measured. And then what changes of other cytokines happening, that's we measured. That's a mock means negative, mock neutral negative. Here is another control, two negative control. Another Nipah virus, another thing, okay? And then measure all those cytokines. And we saw when there is a neutrophil, neutrophil, neutrophil. So with neutrophil and without neutrophil, okay? What is with neutrophil, there's inflammation, more thing happening. So that's just to say that it is happening on human. So in human, when they got infection, somehow the neutrophil coming different circulations, go to the lung, excuse me, and then it induces more inflammations. Like you can see those MCSF or GCSF, all those increase, and that's inflammation happen and the pathogenesis happening. So this is a nice model, not only for Nipah virus, any disease you can do it. So this is a summary. Oh, so basically what I summarized, it's there. Okay. So this is the team we did in NIH. They're all in BSL-4 suit. So this is the chips we used. So there are a lot of collaborators in this projects. And also I've not shown the vaccine study, what we did, so that we need a CDC's permission to show, so we have not done. Okay, so next. I think okay, yeah. No. TNF alpha is produced by immune cells, okay? In organochip, we have not put any immune cells, right, other than neutrophil, right? Theoretically, what happens when the virus there are a lot of immune cells there, and the immune cells will produce TNF alpha. It is a cytokine, okay? It's a cytokine, it's a protein. I think it's a protein, and it produces, and it does a lot of effect. It's mostly killing cells. Okay? So when you have is produced, the function is TNF alpha to kill that infected cells. Because in organochip, we don't have any neutrophil, we have not put any lymphoid cells, okay? But only we put the neutrophil. Neutrophil also produces TNF alpha too. Just to mimic, because organochip has no other lymphoid cells, only the neutrophil. That's why you put externally TNF alpha, imagining that When the other cells are there, they are producing already. What happens? Why the TNF alpha is there? Is that answer your question? Okay. So always think about like all those cytokines increase. They have majority is inflammation. They increase the inflammation. How they increase the inflammation? They recruit neutrophil, monocyte, platelets. That's why they ask everybody to come here to do something. And then when they come, they either do clotting, either do those bad things. Okay? So that's why, always yesterday we were saying, like, you have to reduce the inflammation first. At some level inflammation is good. Otherwise, without inflammation, also not good. Right? Some level of inflammation is good. If it is too much inflammation, is bad for your body. And also, inflammation is also important at some level because it will induce adaptive immunity. Because inflammation comes by, it's the innate immunity. Okay, so Marburg, because we talked yesterday about Ebola. So Ebola, Marburg, they are very close. They are like buddies. But it is not like, it's a highly pathogenic virus, but it's not like that much transmissible. There are very few incidents happens, but when it happened, there is a death there, right? You can see it started in 1967. You can read all those, like in Germany, 31 laboratory workers infected, 7 dies. 1975, 2 travelers, 1 nurse were infected, 1 died. So far, every year kind of, you can see those. Mostly people are traveling to Africa. They are died here. Like one traveler returned from Uganda was infected. One traveler returned from Uganda is infected and he later died. But otherwise, it's mostly prevalent in African countries. And there is a lot of deaths there too. And again, like Ebola, it's same like elongated virus. It's very big. Same proteins like Ebola has. They have everything because they are all related to the filoviridae. And the two important is the Raven virus and Marburg virus. So they are the reservoirs. Excuse me. So the Egyptian fruit bats, they only they survive with the fruits only, and they are also the same kind of bats. So they are the major reservoirs. Transmission happens from bats or some intermediate hosts, mostly NHPs and bushmeat. So bushmeat is I told you HIV is another cause like bushmeat, right? HIV was there in monkeys. I mean, it was SIV, they call simian immunodeficiency virus. It was in monkeys, and then people, use the monkeys as a meat. They cut it and eat it, but if you don't cook it properly, sometimes they eat raw, and it transmits to the human, and then there are more mutation happens, and the virus adapted to the human and becomes a huge epidemic of HIV. That is the theory which is my previous mentor told, and he published a lot of paper on Nature and Science altogether. So the same thing here plays the same thing, like bats goes to the non-human primates, and in Africa a lot of people eats those monkey meats, and that is another way of transmission happening. So this is another electron microscopy pictures by you can see after infecting cells, those are like elongated virus are coming out, and this is another. It is very coiled form. It's only 2 micrometer long, but it is a highly magnification, like 140,000 magnification. So what happened to the virus-host interaction and in man? So the susceptible persons, they have a lesion, skin or mucous membrane, nose conjunctiva, the primary infection happens. Infection happens also some patients, so they have not too much going on those, but if there's more systemic disease happens, so it just they recover somehow. That's why you've seen like now 10 people got infection, only 1 died or 5 died, so what about the rest? So they are like this. They don't show too much. Their immune system maybe kicked in some point. But those that does not, they have the systemic disease infections. and it come out. It also, goes to recovery stage, and also it may be always present those, as like Ebola on the liver, as well as in the semen, and also it's excrete to urine and all those things. Even if they have a hemorrhage, it comes to the blood too. So this is the timeline for this. You get day 1, high fever, high headache. Then you have a diarrhea, day 3, abdominal pain. Then flu-like symptoms, day 4, day 5 and 6, you have a hemorrhagic manifestation. You are bleeding everywhere, okay? And by day 13 onwards, you got a shock because you have a loss of blood, loss of fluid. You have a lot of clotting happening in your body, and due to shock, you die. So this is the thing. It's kind of summary of what we talked about. From bats and NHP, the human got it, and they saw all those infected organs. They see all those things. The fibrin thrombus, because it's also go to the brain, causes encephalitis. And then it causes tissue necrosis. It goes to the kidney. That's why it come out through the urine. It affects the gastrointestinal tract. That's why they have a diarrhea. And the application I think we talked about with Ebola is the same thing, the virus. So here the receptor is this, NPC1 by DC sign. DC sign has another cells. It's a dendritic cells. They express the NPC1. So when the receptor detect those viruses, mostly like a glycoprotein, it binds to the glycoprotein. It can get encapsulated here. And then all those translational happens using the endoplasmic reticulum, and then packaging happen, the Golgi. So then they, packaging, after packaging, release from the cells and the new this is the diagnostics antibody captured ELISA, RT-PCR, definitely need BSL-4 labs. There is vaccines available, and all this treatment is very supportive, like Ebola we discussed yesterday. Fluid therapy, mostly fluid therapy, and any symptomatic treatment. If you have a headache, give some painkiller or something, those things. If you have some secondary infection, they give some antibiotics. Candidates still are under trial and to evaluate how the efficacy of those. So it's nothing foolproof and recipes there available for those Marburg, but the like there are antibodies available, especially or old people, but it's not. Okay, so you should look, that lady, she's not here. Oh, you're here. So Bats Conservational International. So you should look over, they have a nice website. Viruses, especially Hendra, Nipah, Lassa viruses, Marburg. All those bat species are involved for the transmission, okay? And they are localized in those countries. So you can imagine, how much bat harbored those viruses, and they're able to transmit to human, through that route. They we saw Nipah, like bat, human, or maybe direct. Australia, I know there are a lot of studies going on from this because it's so many families there in Australia, and they are doing. You can see, they mentioned, I never went to Australia, but they said if you go in the street, you can see the bats are there in the road or somewhere. those fecal samples and do the different kind of microbiomes. Really like how much pathogen is urine or fecal samples. So yeah, like more bat. You see, bat's evolutionary age is 64 million years ago. There are a number of species so far. They see 1,122. Okay, it's a huge the distribution. Every but everywhere. Interesting. I like to say like they those things. Uh, Reproductive size bimodal or seasonal and aseasonal means bimodal means they sometimes they can switch. So another interesting their lifespan maybe more than 41 years or above. So another thing I like to say, so there yeah 10 to 16 bits per minute, but when they're flying it comes to 1000 bits. So and they have a high metabolic rate also because when they measure the oxphos genes are actually um, it is a pathway where body the cell produces ATP. We need all ATP. So that is the energy source. So they have a lot of energy source. If their metabolic rate increase 34% when 34 times when that history here because no doubt they are able to transmit Africa, and Africa is a huge source of different because other countries, right? We cut the trees, we all do this, so make huge, tall buildings, so the bats cannot live. Other things, what is the bats doing? Interested. So why, a lot of questions came, why bat has a lot of virus, but they are not get infected. They control the innate functions. Uh, but in human, they are all upregulated. So the innate what is that does, like there are a lot of body receptors like TLR, NLR, CLR. Those are actually pathogens. They bind to those damp. Those are the particles from any viruses, any bacteria, any pathogens. Those TLRs, NLRs, CLRs bind, and they increase this kind of cycle where they induces all those functions. And that is very common. Any animals, humans, here they are comparing humans. That's why bat gets a lot of viruses or pathogens, nothing happened because they switch up all those innate immune functions and they can harbor those viruses. So if you switch up innate function, you cannot generate an adaptive immune response. So no antibody production against this pathogen. They're cutting those. There is no immune function against. So they say it is a self. It is a not foreign. If you switch off those, it is like a self antigen, not a foreign antigen anymore.
No, I got that the immune system is stopped from responding.
Yeah.
But the virus itself can replicate and
Yeah, virus replicating, then no problem. But it's not killing the cells. It is infecting another cells. It's not killing the cells. So this virus is replicating, virus is shedding. Every day virus is shedding, no problem. Check the adaptive also, because if the innate does not kick in, adaptive cannot do anything. So that's the major function. The bat has a lot. And another thing I like to point out, there is a huge study going on to the sequencing of all bats. By 2040, they will complete everything. You can go to the website they have. There are a lot of they NCBI, and then a lot of papers can be done on this. Right? So, yeah, and I'll stop here. Take over.
how is this bat not dying when when the virus is infecting one of its cells? It's not defending back in any way.
No,
Why is it?
so, switch up those innate. That then it is like a self antigen. So you never trigger. Do you trigger a antibody in your own cells? No, we don't, because it's a self antigen. They consider it as a self antigen.
Let's say my immune system does.
That needs further study. Well, your question is genuine, because if we in fact not doing this, yeah, I don't know,
So, for
because
example, if the same thing happened to an immunodeficient person in this,
you cannot compare with immune system deficient with this. Immune system deficient people will never live. They'll be not doing that way. They are just stopping some key sections which is not important, which that can help. You have to do more research. You have theoretically the experimental design will be you take what is that, pteropoid you isolate cells or do different assays, maybe next generation sequencing and what change is happening in the gene sequence. So regulating or down regulating, then you can do more cellular functions. You can isolate those cells, do in vitro cultures in presence of virus or without, and see what change is happening. That way it will be fine-tuning the mechanism of it, why bat cannot kill those cells like what happened in humans, but they are harboring those virus forever and ever.
am I right in understanding that our immune system defending us in case of Nipah and other viruses is us?
No. See, basic principle is when you get an infection, your body thinks that is a foreign. We told you, right? Body will interact. What will come first? Innate comes first. Without innate, adaptive will not come. Okay? Innate comes first, it induces inflammation, it it recruits neutrophil, platelets, and then when it comes, the neutrophil also, macrophage comes also. Macrophage also come at the same time, and then in engulf, antigen presentation happens, adaptive immune system come. That is the normal theory. Nipah virus, what happens for human? The same thing. Innate function increase, that's why you see fever and vomition, all those things. All the effect of the pro-inflammatory cytokines. Like COVID also the same thing. Every virus the same thing. So they, and then eventually the antigen presentation happen, you get a nice antibody and you protect. Some people can handle it, some people, if you don't treat it, they will die. Okay, because everybody's, human is heterogeneous, everybody has a different thing. So I may die, but this guy may survive because he's better immune system. He has maybe better good memory. Okay, maybe I have some diabetes or something. My immune system is very shaky in that aspect. But bat is different. Bat initially, they after infection, they stop those innate function. Yeah. But what happened to the macrophage? I don't know. Nobody has studied that. What happened to the antigen presentations? I don't know. So those are the things. But seems like they are also defective because otherwise, or maybe it may true, maybe they may generate a lot of antibodies. You know, I am thinking otherwise. Because the macrophage is also functioning easily. They generate a lot of antibodies, but they are also checking the virus. But it's not happening for Nipah because Nipah, they're all continuously shedding the virus through urine, through fecal samples and all those. So it seems like the bat does not generate enough antibody or T cell response to control the virus. The virus is multiplying. Same like monkeys happened. We talk. Because we don't know. Yeah, we don't know those things. Because that's the difference. Natural host functions of immune function. The African green monkey, they can take the HIV fever. Initially they go down, but they are fine. That's why they saw natural host. All those viruses we talk about, natural host means their immune system cannot do anything on that virus. They can harbor. Like in your gut, there are a lot of bacteria there. I can say, oh, why the bacteria? I should have antibody and get rid of it. No, they are natural. And they are needed for it. Exactly, they are needed for it. We need them, otherwise we'll be dead. So they are natural. I am natural host for them, for that bacteria, that viruses. But not for Nipah, because if Nipah comes, Nipah will kill me. But that bacteria will not kill me. Like Candida, we talked about, we have a Candida, everybody has Candida. But if I have immune suppressed, the Candida will kill me. But then Candida will be very pathogenic. But normally Candida is there, you know, it's under control, everything is under control. Yeah, it's a balance, you know. Exactly, yeah.
But still, they won't die.
Don't die, yeah.
There might be some
Yeah,
mechanism there.
yeah, yeah. So this young generation should do some more study, and we like to.
Can you show us that paper? Can I get the reference?
I cannot control those guys. Well, yeah, I'll show you. Yeah. No, she want to see the previous slide, the reference. So.
He has a question.
Yeah, sorry. Go ahead. Huh? Right, yeah. Right. There's a lot of studies done, P53, yeah. Yeah, probably. There are a lot which has not been studied. Like, because nobody cares about, Ebola, Marburg, Africa. If it happened to USA or something, maybe. So definitely those studies are missing, still missing. That's why people are initially doing why fruit bat is different than the other bats, and other bats are having the Ebola. So those to short. Then those, so many pathways like P53, NF-kappa B pathway, MAP kinase. So there are a lot of, those are all related to also cancers and other disease, and very important. So yeah, those need to be explored because I don't think there is any like systematic studies has been done or designed for it. So yeah, I don't answer because I never studied bat immunology, but I know I went to the Keystone conference, I think last year, Germany. There is a huge group working on those biology, and they are doing all those kind of. They have the lab to do that. They keep the bats. They do possible for a one. Everybody has a different kind of. Yeah. Okay. Not failing. I mean, aging immunology is a different subject also. Yeah. Go ahead.
Yeah, so in bats also, that maybe at the young age they would be focusing on
Yeah, yeah. No, we don't know. That's why we don't know because what age that bat is more transmissible than the other age, right? And whether it is more older, they are transmit easily, or it's younger is transmitting easily. So yeah, we have
itself.
right. So,
Any further questions? You can have.
yep.